have another interesting edition of Youth Mental Health Perspective Seminar Series, starting this academic year with a very exciting speaker. I'm very pleased to introduce Dr. Jibran Khokhar. On Zoom, Jibran, we have around 20 people, so it's recorded as well. Information. So Jibran needs no introduction for people doing neuroscience in Canada. But I have a few bits I really want to highlight. And before I even read out any of his laurels, I want to introduce him by saying, you will not have a more nice speaker in terms of nice, how nice somebody is in supervision, in collaboration, in co-investigation, in just being a colleague. Jibran is one of the nicest that I've ever had the privilege to work with. So I'm very happy for Jibran to open our current academic year's perspective series. Jibran did his undergraduate training at Queen's, not far from here, and his Ph.D. at Toronto in Pharmacology and Toxicology at CAMH with Dr. Rachel Tyndale. He then completed a Post-doctoral Fellowship in Dartmouth with Dr. Alan Green. And Dr. Khokhar, Jibran, held a CIHR Post-doctoral Fellowship as well as a NIH K99 R00 Pathway to Independence Award from National Institute on Alcohol Abuse and Alcoholism. Now, those awards are very hard also for international graduates to get in the US. So an exciting start to his career. And he was recently hired as an associate professor and Canada Research Chair in Translational Neuropsychopharmacology in the Department of Anatomy and Cell Biology at Schulich School of Medicine at Western. Since starting his independent research group, the lab has published 53 papers and received nearly $6 million in funding from federal and industry sources. Dr. Khokhar's research revolves around establishing the behavioral and neural correlates of various types of vulnerabilities to substance use, starting from genetic risk to effects on development. He's also very interested in pharmacokinetics of various routes of drug exposure and has established tools and methods to study emerging routes of administration, such as vaping and edibles. And his talk today is going to be on vaping, and it's particularly interesting and exciting for our group because we're working on emerging symptoms of youth with mental health problems. And before symptoms emerge, before problems emerge, the one thing that emerges is vaping. So you're going to tell us more about vaping. Thank you very much, Jibran.
Thank you. And what I can say about Lena is it takes one to know one. And so the nice thing applies just as much to him as it does to me. And so thank you all. And thank you all for being here. I'm excited to be able to share some of our research here with you today. But before I start, I'd like to acknowledge that the work I'm about to present was performed both in London and in Guelph on the traditional lands of the Misagas of the Credit First Nation of the Anishinaabe people, the Haudenosaunee, the Lenape-Awak, and the Chinookan nations on lands connected with the Dish with One Spoon Covenant Wampum. And as a neuroscientist, or as neuroscientists, while we try to figure out the most complex mysteries of this thing up here, we must recognize that neuroscience is done by flawed human beings, us, and has been and continues to be used to uphold systems of racism and colonialism. And this is not something we should shy away from, but instead see how we can contribute to both understanding and mitigating the long-term consequences of settler colonialism and genocide on the brain, body, and society here in Canada and across the globe. And this is where, in the context of substance use, especially in adolescence, I think we have an important role to play even there when it comes to decolonization and understanding the impacts, especially on Indigenous communities. Substance use in adolescence is an important topic, and we could talk about the numbers and how it's a pediatric condition and how it starts. But for me, the way I think about it being important is how often us adults try to scare kids out of using drugs, right? Like, so going all the way back to Nancy Reagan, this is your brain on drugs. Fried eggs don't really have much to do with drugs unless that's what you want after you use cannabis or something else, and that's okay, that's your choice. And that hasn't changed. We still continue to try to use the same methods, or sometimes misinformation or disinformation, to try to scare kids out of using drugs. And that's the same with vaping, where they're trying to now use scary images. Vaping can put dangerous chemicals like diacetyl into your lungs. And maybe that's true, but that doesn't work. Just Say No didn't work. The DARE campaign didn't work. In fact, if anything, the folks that were enrolled in the DARE program ended up using drugs more than the people who weren't enrolled in the DARE program. So it's about time we try to figure out, how do we talk to youth about drugs, and how do we tell them about the effects of these drugs? And so the goal when we started the lab was to try to see if we can, instead of misinformation, use an information-based approach and see if we can maybe use animal models to predict what might happen, and then try to disseminate this information in a way that was accessible so that the youth can make informed decisions about their own substance use. So as far as trends go, things are looking actually pretty good. So this is Ontario data from the highs in the 70s to the 80s. Alcohol use is down, tobacco cigarette use is down. Cannabis for Canadians probably will never go down. Right. That's but it hasn't gone up since legalization either, and that's what's important as well. In the U.S., the data looks similar. Past year alcohol use in grade 8th, 10th, and 12th graders is down. Cigarette smoking is also down in the same grades in the past from 2009 to 2019. I have specifically not used data past 2019 because COVID threw a wrench into the works, and everything is still fluttering because of it. And so we'll see in the next couple of years how that recovers. This is then data, but not all of it is going down in this direction. This is data from the Ontario Student Drug Use and Health Survey. We see increases and almost a doubling of vaping use. This is nicotine vaping in males and females, and this is all in 12th graders. And then from the US data, it's pretty much the same, where you're seeing doubling in the nicotine vaping and the THC vaping rates in the Monitoring the Future survey. And even the latest release that came out in 2023 of the Monitoring the Future survey suggests that these rates have pretty much remained that high since then. So our lab has been, especially in the context of youth mental health, what we're talking about here, and all of your research interests, our lab has been interested in trying to figure out what, if anything, is causal in the associations that we see in human studies. So we have longitudinal studies, we have cross-sectional studies in humans using substances during adolescence. And the data there suggests that adolescence is a unique period where if somebody uses substances during this time, it puts them at a greater risk for basically three major domains. The future risk for developing a substance use disorder, where we see the earlier you start, the greater your risk. The later you start, the lower your risk. The other one is psychopathology, and it's a chicken or egg question that we've been trying to tackle in the lab. Increased risk for schizophrenia, depending on what drug it is. Cannabis, for example, increased risk for anxiety, bipolar disorder, depression, depending on which drug we're talking about. That's another domain. And then the last one is cognitive deficits. And so what we want to figure out is any of this actually causal, or can it be explained by other factors that we're just not accounting for? And is there an inflection point that which or before which, if we intervene, can we reverse this in some way or form? And so that's where we're at, and that's what we're hoping to do with the animal models and the reverse translational approach that we've been taking in the lab. Rats are not humans. Mice are not humans either. But they can be useful heuristics that can allow us to at least study the fundamental mechanisms where there is conserved neural circuits that might be the same. And because of their shorter lifespan, it allows us to study a period like adolescence in 14 days. So day 28 to 42 in a rodent is the entire adolescent period. And by day 60, they're adults already. So another, 18 days and you're already in adulthood. And that's a period that's short enough that allows us to actually do these studies and see what is changing if the only thing that's changing during their developmental course is whether a drug was present or not. And how does that brain change? And what we've been doing is, and by no means am I the first or the last to do studies like this. We stand on the shoulders of giants like Linda Spear and others who have really, first characterized that this is the lifespan of a rat, and this is where the, the correlating ages are. But one thing that we've tried to do that's slightly different is both mimic the exposure in humans, maybe better, and mimic the outcome measure in a way that's translatable. So by using vaping, for example, as the exposure instead of injected nicotine or injected cannabis. And then as far as the studying of the brain on the other side, using translational preclinical MRI imaging to study the brain so that the changes that we detect in those brains may be translatable, or at least in a domain or in a signal that's similar enough to what we can actually get in humans as well. And so some of those tools that we've developed, one of these is an open source tool called OpenVape. You can build it for $250. Basically, it's an Arduino that's driving pumps that can be attached to anything. In this case, this is a Juul cartridge. Juul was the biggest player in the market until it was scapegoated and taken out of the market, only for many things that are Juul lookalikes to pop up and still be present. And then the governments feel like they did enough and Juul's gone, but everybody's still vaping. And so what we can do here is control the number of puffs that animals get, how long for, and you basically hotbox the animals with the vapor from the cartridges, and that's how they get exposed. And so in the first study, we looked at whether, just like in adolescent humans, Juul vapor was actually rewarding or not. Initially, when the Juul e-cigarettes came out, the manufacturers actually said that they were not addictive. In fact, I remember the first time I heard about Juul was actually back at Dartmouth College. I gave a talk at a sorority once, and the talk was all about alcohol. It was not about nicotine at all. But as soon as the talk ended, all the questions were about Juuls. And I said, I don't even know what a Juul is. This was in 2016. And I said, I don't know what a Juul is. And they said, oh, these are these flavor pods that create vapor. They have no nicotine. The truth couldn't be farther from that statement, because Juuls actually changed the market by using a different kind of nicotine, a nicotine salt, and a higher dose of nicotine than any other e-cigarette that was available on the market. And so we set out to figure out if, in fact, this vapor is rewarding or not. And this is a paradigm called a conditioned place preference design, or a paradigm in which an animal is first asked to see if two chambers that are different in terms of the grates, the smells, the patterns on the wall, if either of them are preferred by the rat. And then what we do is take the chamber that was not preferred, the place they didn't like initially, and see if we now expose them to Juul vapor in that one, in that chamber, does their preference change, right? Like, because now the Juul is going to make them feel a certain way, and that might then make them associate it with this chamber. And so we did this, over a period of 7 days, and they were either exposed to nicotine Juul vapor or just the propylene glycol and vegetable glycerin that is in the Juul e-cigarette, and that was done on the side that they initially preferred. And what we find is that both adult males and adolescent females, adult males and adult females find short durations of exposure. So 480 seconds, or so up to 10 minutes of exposure once a day to be rewarding. What's interesting is adolescent males actually find the same doses to be more rewarding compared to the adult males, but the adolescent females actually don't find nicotine vapor from a Juul e-cigarette rewarding at all. And so our lab is trying to now figure out whether it's something related to stress and stress relief. And so we're trying to do the same experiment now with stressed adolescent females and seeing if that then reverses or changes how rewarding they find nicotine to be. The other thing we looked at, and again, to test those initial claims by Juul manufacturers of it not being addictive, is whether animals show withdrawal symptoms after nicotine or Juul vapor exposure. And in this case, animals were exposed 10 minutes for 10 minutes, 3 times a day for 14 days. And then withdrawal was precipitated in this case by giving an antagonist of the nicotinic acetylcholine receptor. What an antagonist does is it blocks that receptor where nicotine usually binds to, and as a result, the animal feels like there's no nicotine anymore, and so it goes into a state of withdrawal. And you can measure that by looking at somatic signs. Some of these somatic signs that can be seen in animals after nicotine withdrawal include teeth chattering, head shakes, yawns, ptosis, foot licks, cheek tremors, eye blinks, etc. And you can basically record video of these animals and score it manually, but also using computer vision to assess what are those differences in how much withdrawal these animals are showing. And what we see is that both male adult animals and adolescent animals show significant withdrawal when exposed to nicotine compared to the control groups, but female rats, both adult and adolescent rats, do not show any withdrawal. So we're seeing these emerging sex differences that are happening both in the rewarding effects of Juul vapor and withdrawal. What's interesting is we wanted to figure out what is it that causes it. And so we looked at the pharmacokinetics of it, and we see that even though the male adult and adolescent animals are, the adults are 3 times the weight of the adolescent animals, hotboxing results in the same plasma nicotine levels in both male, adult, and adolescent animals. Where we see an interesting difference is the female adults have much higher levels of nicotine in the plasma at the earlier time point compared to the adolescents. But the biggest difference we see is the sex difference between the male and the female animals, with the female animals resulting in much higher levels of nicotine. An important point here is that if we ever gave an injection of nicotine that resulted in this high levels of plasma nicotine, the animals would probably be dead. Or at least gasping and find it hugely aversive. But because it's a nicotine salt and it's a vapor, the animals actually find it rewarding, like we saw previously on the CPP slide. And so it's important that we mimic the routes of administration that humans use in a way that's actually relevant. And the outcomes would be very different based on the route of administration used. Similarly, we also see the same difference in the brain nicotine levels between the male and the female, and that's also seen in the adolescent male and female animals as well. And so this might explain some of the differences that we're seeing in these animals. We also wanted to see what happens in the brain, and much like Malar here, we turned to preclinical MRI. And I always include a Zoolander reference in my talks because when I talk to the kids in classes these days, nobody knows what Zoolander is, and that's a shame. So I'm going to change that one talk at a time. But yes, so we do have a MRI for rats. It's a 944 Tesla MRI that we've been using to do these studies at Western. The animals were exposed the same way that the previous study, the withdrawal study and the PK study, animals 3 times a day for 10 minutes, 14 days, either during adolescence, 30 to 43, or in adulthood, 74 to 87, and then scanned 2 weeks later under a combination of isoflurane and dexmedetomidine anesthesia, which preserves neurovascular coupling and most of the resting state networks that can be seen in the rat brain. And we collected both anatomical, diffusion, and functional data, and that's what it looks like in the rat brain. And here we're using an approach called network-based statistics. And again, using tools put out by the Cobra Lab for pre-processing, in this case, using rabies, the rabies pipeline. Here we see that nicotine-treated animals show reduced connectivity compared to the control treated animals across a pretty broad network, which includes, so this is network-based statistics is a graph theory based assessment or statistical assessment that treats individual brain regions or functional brain regions in the atlas as nodes, and every connection between 2 nodes is considered as an edge. And we see that 12 edges and 13 nodes showed reduced connectivity in nicotine-treated animals in a lot of the same brain regions that actually also show reduced connectivity in humans, especially human smokers, including the insular cortex, the cingulate cortex, the hypothalamus, the piriform cortex, and the hippocampus. So not only are the behavioral findings, consistent with the humans, at least even the brain, and so, while rats are not humans, if it walks like a duck, talks like a duck, and the brain scan looks like a duck, then maybe it's not a bad model for the duck. And that's what we're hoping to continue doing with these findings. But what's really interesting is, unlike the behavioral studies, where the females weren't really showing much of anything, here we see a sex by group or treatment group interaction, with the females having significantly lower or reduced connectivity in a smaller network, 5 edges and 6 nodes, the hypothalamus, piriform cortex, the CA1, and the auditory and visual cortex, compared to the males. And so how we're starting to reconcile this is the females had much higher levels of nicotine. Nicotine, while it acts on the nicotinic acetylcholine receptor as a ligand, also acts as quickly desensitizes those receptors as well. And that's possibly why we weren't seeing a behavioral signal, but that might be resulting, and because the scans were done 2 weeks after, when the nicotine is gone from the system, we might be seeing a greater effect in the female rodents compared to the males. Now turning our attention to cannabis. Cannabis needs no introduction in Canada. We remain the highest users of cannabis worldwide. And again, just like the previous slide, we've taken a very similar approach to talking to kids about cannabis as well. I don't know if anybody's heard of this campaign, but I'd recommend checking it out on YouTube. It's called Stoner Sloth. It was out of Australia. Again, if anybody's seen Zootopia, it's like the sloth scene from Zootopia. That's what they're basically telling kids, that this is what cannabis will do to you, which is, again, not based in evidence. In fact, there's something called the Nesbitt's paradox when it comes to cannabis, whereas it might slow processing time or reaction time, it actually increases impulsivity. And so it might balance out in the end, and you're not any slower in terms of your reaction time. And so it's important that, again, we lead with information instead of misinformation. Cannabis use in Canada, past 12 months use, over a quarter of Canadians use cannabis, but especially when you get into that 16 to 19 or 20 to 24 demographic, that's almost more than a third, and 50% of Canadians report using it. And what's also important, and this is Stats Canada data, and so, Stats Canada can be useful sometimes. There, you see, like, even, if we were talking about nicotine e-cigarette, and its popularity has largely been suggested to be associated with its safety profile. It's safer than e-cigarettes, which is a low bar. Everything's safer than regular combustible tobacco, right? But even compared to nicotine e-cigarettes or alcohol, which is, ubiquitous in our society, cannabis across its various forms, vaping, smoking, or eating cannabis, is perceived to be safer by Canadians compared to those other drugs. And so it's, again, important that we try to figure out what that safety profile actually looks like, and is there truth to this perception or not. And so, again, trying to mimic human exposure as best as we can, we turn to the OG vaporizer. It's called a volcano vaporizer. It's a tabletop vaporizer. Usually when people used to use it pre-COVID, at least, a balloon would fill up with cannabis vapor, and then you would puff pass it. Post-COVID, we don't share the balloon, so they've created what's called a whip attachment, and we can just take the whip attachment and attach it to a cage in which there's 16 adolescent rats that are hanging out, and you can hotbox them with cannabis vapor. And so we went to the Ontario Cannabis Store, and you looked at the Yelp reviews. What is popular? What kind of cannabis are people buying? And so in this case, this is called Trudeau Wedding Mint. 5 stars, has one of the highest amounts of THC, at least in a flower, available on the cannabis store. There's the Tweed Balance, which has around 10% THC and 10% CBD. And then Pure Sun Farm CBD, which has around 20% CBD. I don't know why anybody would use that, but people apparently like it. And so adolescents were exposed from postnatal day 28 to 42, and then in adulthood, they were put through a variety of behavior as well as MRI assessments. And for the most part, and it's really important whenever we're doing vapor exposure studies, unlike injections where you know that the drug is getting in, vapor exposure, the animal still has volitional control. They can decide either to go into the opposite corner and stick their nose in the corner, reduce their breathing rate, or they can go and stick their nose right where the vapor is coming out and get the hit directly. And so it's important to then anchor it to something real. In this case, it's important to anchor it to both pharmacokinetics. So what are the concentrations of THC and CBD or the primary metabolite, but also behavioral measures. And for the most part, the Ontario Cannabis Store and the labels on that cannabis are not lying. The high THC cannabis had the highest plasma THC levels. The high CBD cannabis had the highest CBD levels, and the high THC cannabis again had the highest 11-hydroxy THC levels, which is the primary active metabolite of cannabis, of THC. And then behaviorally as well, we see that the high THC cannabis results in an increase in the tail flick latency, changes body temperature, and reduces motor activity as well. So then that was all when the animals were being exposed during adolescence. We let the animals grow up into adulthood and tested them on a variety of learning paradigms, both in relation to that substance use risk we were talking about earlier, but also in relation to the cognitive deficits that have been observed after adolescent exposure. And we see, so this is what's called a sign tracking task. Basically, it's a box with 2 levers and a food reward port. Lever comes out, lever goes back in. Regardless of whether the animal presses the lever or not, a food pellet is delivered. But over time, animals form an appetitive association between the lever and the food pellet, where they'll start to gnaw at, bite at the lever at every opportunity they get. It's kind of like the Golden Arches and the McDonald's, right? For us, the Golden Arches have never given us a Big Mac, but every time you drive by, you notice the Golden Arches and you might think of a Big Mac and might pull into the drive-through. And so what you see here, the control group learns that association pretty well, where by day 5 or day 6, they're pressing the lever maximally and learn that pretty well. The balanced cannabis group actually doesn't look very different from the control group. And so, no real changes in reward learning in these animals or cue reactivity. That sign tracking can also be used as a measure of a reward cue reactivity. What we're seeing is in both the high THC and the high CBD groups, they actually never learn that to the same extent or in that appetitive learning task. When we move to an aversive learning task, in this case, it's called conditioned avoidance response. Basically, light comes on. If the animal escapes to the other side, it can avoid being shocked, but if it doesn't, it will receive a shock. And over time, you see the control animals, this is in a single day, 100 trials, so 10 trial blocks. The control animals learn this really well, where 80% of the time they're effectively avoiding being shocked. Compared to the controls, all 3 types of cannabis, whether it was high CBD, high THC, or balanced, don't learn this aversive task nearly as well. Yes, Milan. Yeah.
That's really interesting, but I guess the question I have is the balance was 10%.
Yeah.
Right? So what if you had a 20%?
Yeah, no, and that's where the limit of using products available on the market is. When you're, and this is through selective breeding, when you try to either increase one, the other one goes down, and you increase the other one goes down. So we couldn't find a product that had 20/20. The other thing that we did try is doubling the amount of flower, but that didn't scale. And so the plasma levels were not twice as high when you double the amount of flower. So we're still trying to figure out, but that's one of the limitations of using products that are commercially available.
I guess maybe you get this in a second. Were you surprised that the high CBD THC groups performed the same?
Yeah, no, we thought that either the balanced, and maybe when we first saw the appetitive data, we were like, okay, that's what we would have predicted. CBD undoes the harm of THC, right? But then both the appetitive study and the aversive, the high CBD group is showing the same thing. And so it might be CBD itself, or it might be that any presence of THC, whatever the dose, is enough. Which, if you think back to like Hugh Garavan's study with a single exposure, and we're about to show some other data that even you, Malara, and Rachel haven't seen from the last talk either. A single exposure to cannabis during adolescence can have long-term behavioral effects as well. And so maybe any THC is enough.
What is the THC in this
CBD group?
It was around 2 to 3%. 2 to 3%. An important condition here, though, is in a model that we often use to, so just like on that first slide, I had substance use risk, cognitive deficits, and the third one was psychopathology. One model that's often used to model psychopathology in rodents is what's called pre-pulse inhibition of the acoustic startle response. It's disrupted in patients with schizophrenia, patients with bipolar disorder, and it's one of the more robust findings that we have with THC in animals. If you give injected THC to animals, it reliably disrupts pre-pulse inhibition in adulthood, but not when you vaporize cannabis, and especially give it at the low levels that we were giving it at, right? Like our plasma levels were anywhere around 5 to 10 nanograms per mil. When you inject an animal with even a low dose of THC, you're in the 100, 150 nanograms per mil. So there's a big dose difference, but at least at this low dose, it does not disrupt PPI across any of the types of cannabis used or across any of the pre-pulse intensities. And so now going to the brain, we do see some interesting and important differences. And this is with functional connectivity, again, using that NBS approach. We see two networks, one network in which there was significantly higher functional connectivity in the CBD group compared to the control group. And then another one where we see higher functional connectivity in the CBD group compared to the THC group. And it's very interesting targets. So somatosensory cortex, primary and secondary visual cortex, retrosplenial system, raphe median pontine reticular nucleus. The pontine reticular nucleus is really interesting, is because that is the part where PPI in the brain is mediated through. And so maybe we weren't looking at the right behaviors. And so now we're actually changing in collaboration with Brian Ullman, which Lena, you might know. We're actually now looking at audiovisual related and somatosensory related behaviors to see if these behaviors now show differences, but also at the same time use machine learning to see what features in the NBS data actually predict the behavioral outcomes that we're seeing. When we look at structural connectivity, some of the usual suspects show up. And so this here, we see significantly lower streamline counts in the control group compared to the CBD balanced and the THC group. So there's increased connections in the three cannabis groups between regions, including the insular cortex, the striatum, the ventral thalamus, and the endopiriform cortex. So regions that mediate, addictive potential or addiction-like behaviors, there's increased structural connectivity in. But when we look at brain regions, when we look at other brain regions like the entorhinal cortex, the interpeduncular nucleus, pontine nucleus, and the raphe median or reticular nucleus, we actually see decreased streamline counts in all three cannabis treated groups compared to the controls. All the data I presented so far was in male rats. When we look at female rats, they're not affected by cannabis vapor in adolescence at all. In the sign tracking task, we see no differences between the groups. And even in the imaging, we actually saw no significant differences between any of the cannabis-related exposed groups versus the controls.
What do the female rats do when the exposure happens? Do they hide away? Do they run away?
They show the same variability. Like some will go and stick their nose in and others will go hide away. But the interesting thing is when we look at the pharmacokinetics, those labels on the Ontario Cannabis Store no longer hold true. The females are much more efficient, and it's because, especially when it comes to female adolescent animals, they have lower body weight, but they also have lower body fat percentage compared to male adolescents. And so the volume of distribution for these lipophilic compounds is very different. And so even in that high CBD group, you can see that the females have significantly higher levels of THC, even from that high CBD group. They achieve higher levels of THC with the high THC group as well. But even with the high THC group, now these animals, the females have much higher levels of CBD as well. So they're not getting the same drug. And even when it comes to metabolism, they're so much more efficient compared to the males at converting that THC into the 11-hydroxy THC. Yes, Malar.
Do you have videos of what the animals are doing?
Yeah. We have all the video. So yeah, so Hakan is actually putting that through Deep Lab Cut right now to try to see if we can both identify, and we have markers on them that we can identify which animal it was, but going forward, that's going to be a part of the job. Yeah. Yes.
You know, like,
Yeah.
I mean, even within,
yeah,
which is normal.
Yeah.
Like if we call
it. Yeah.
So if you want some help spectrumizing your ads.
Yeah, and no, I think that would be a great idea. And it's an important idea, both to look at the videos to see how much they're getting, but even if we're just doing it pharmacokinetics, right? Take the pharmacokinetics in an animal and then look at their behavioral outcomes or look at their network outcomes, right? I think that would be also an important factor in considering or figuring out maybe there are, in fact, two groups in the females, and here maybe there would be one group that's down here and the other group that's up here, and that's why we're not seeing any behavioral differences. So I think that's a very important suggestion. Now I'm going to just add a little bit of data potentially explaining what might be happening here. So we took the exact same cannabis that we exposed the rats to, but in this case exposed mice to, and then took their brains out and sent them away to a collaborator at the University of Victoria, also a collaborator of Millar's, Haley Vaccarelli and Maria Tremblay, and they looked at the microglia, and we think that microglia might be playing an important role in both the sex differences that we're seeing between male and female animals in the effects of adolescent cannabis. But another place that they might be important is the big sex difference that's seen in humans when it comes to the risk for, or the risk associated with cannabis use towards developing schizophrenia later on in life. And so Nora Volkow had a paper just last year where their group showed that males who use cannabis have a threefold higher risk for developing schizophrenia, but this rate did not differ in females between cannabis users and those who didn't use cannabis. And so we think microglia might be playing an important role there in that sex difference. And our very early studies, at least after that exposure to that same high CBD, high THC and balanced cannabis, we actually see that in the males, there's a reduction in the number of microglia in all three types of cannabis exposure compared to the control, which is not seen in the females at all. And so, maybe there is some signal that for the sex difference there. And what's really interesting is that we actually see another measure, which is called fractal dimension, which is the essentially the spread of those microglia, each microglia itself, and which has previously been associated with pathology or an incidence of pathology. So the greater the pathology, the lower the fractal dimension for microglia. And what we see is that the balanced cannabis here has much lower fractal dimension compared to the controls as well as the CBD exposed group. And so we're starting to look at this as possibly one mediating outcome. And that's something, you can only do in animals right now, but hopefully with the development of better PET tracers, we can begin to also study this in humans as well. I have 10 minutes, so we'll end with this. Just like the Juul folks, not saying that Juul isn't addictive or doesn't produce withdrawal, even in the cannabis space, one of the things that's largely debated is whether cannabis results in withdrawal or not. And Alan Budney, who is still at Dartmouth and a good friend of mine, has basically spent his whole career trying to convince people that cannabis withdrawal is real. But if Snoop Dogg can undergo cannabis withdrawal, then who are we to say that cannabis withdrawal isn't real? This ended up being a stunt after, because apparently he didn't actually give up cannabis altogether. He gave up all other types of cannabis except for his own brand. But we set out to see if we can actually establish an animal model of cannabis withdrawal. Previous studies have tried with injected THC, and you can precipitate some level of THC withdrawal in animals, but we wanted to see if vaporized cannabis was actually also produce withdrawal in animals. And so very similarly to the data I showed with the rats with nicotine earlier, if you do 7 days of exposure to vaporized cannabis, that same 20% wedding mint cannabis, and then test them, and then add in the antagonist for the CB1 receptor, rimonabant, you actually see, so firstly here, this is total distance traveled. So cannabis exposure usually results in a reduction in locomotion compared to baseline. When we give rimonabant, that reduction goes away. So you can see that that reduction is CB1 mediated. Again, here with body temperature, you see hypothermia after cannabis vapor exposure. Once the animals receive rimonabant, that hypothermia goes away. Again, telling us that these effects that we see from cannabis vapor are very much mediated by the CB1 receptor and consistent with what previously has been seen with pure THC exposure. What's cool is once the animals receive rimonabant, you actually see an increase in total withdrawal signs. All of those withdrawal signs that I mentioned earlier, we're assessing them here. This is a full overall score, but you can also see increases in blinking and head and body shakes. But what was also important to us is, do you need rimonabant? Do you need to give an antagonist, or would you see spontaneous withdrawal occurring as well? And so in the next study, we actually had 4 groups, and where we had the control group, which just had hot air blown into the cage, where they received rimonabant or saline, and then cannabis vapor, and then rimonabant or saline. And what's really cool is we see the same level of withdrawal in both the THC cannabis exposed group that got saline and rimonabant, with increases in a variety of those somatic signs of withdrawal, suggesting that we don't need the rimonabant, and we can actually have even a model of spontaneous cannabis withdrawal after 7 days of cannabis vapor exposure, which now we can use to both test the neural correlates, but also use to possibly test new medications that might reverse some of the cannabis withdrawal. And so the last thing that we'll talk about today is cannabis edibles. Quebec, you're in Quebec, and Quebec actually, we'll talk about it a little bit later, but Quebec did not legalize cannabis edibles in the same way the rest of the country did. And that actually creates a nice experiment in Canada. But again, you know, just like we try to scare kids out of using drugs, we've also been scaring parents, especially on Halloween forever. When I was a kid, it was like razor blades in your candy, right? Like, check your kids' candy for razor blades. I only check my kids' candy to eat it. That's why I check it. But, you know, that media and law enforcement, which loves to create mass hysteria about all sorts of substances, right? Like we had rainbow fentanyl pills in your, if anybody's gone to a cannabis store and looked at how much cannabis edibles cost, nobody's giving that to your kids. Yeah, but that doesn't mean that the risk isn't real. And so compare in Canada, for example, the pediatric hospitalizations by cannabis poisoning doubled in provinces that legalized cannabis edibles. And so Quebec did legalize cannabis edibles, but only in flavors that kids would not like. So you could have like rutabaga flavored edibles or beet flavored edibles. Like I don't even know which adults would like those, but that's what they made permissible. But in the rest of Canada, it was a free for all. So you can get brownies, you can get cookies, you can get chocolate bars, you can get whatever kind of edible you want. And you had a doubling of that rate in the other provinces compared to Quebec. In the US, everything's bigger, including the number of cases. So 1375% increase in the cases of edible poisoning in the past 5 years in the US. And so we set out to create an animal model of edible poisoning. And so again, trying to mimic the route of administration and exposure as much as possible to the human condition, we turn to Zamnesia, which has a really nice recipe for cannabis infused Nutella. And you can just take Nutella and you can take some cannabis oil, mix it together. And just like the humans love it, the rats love it as well. Except if you mix too much cannabis oil into it. In this case, we took 5 founders cannabis oil. It's a 30-to-1, around 30 milligrams per gram of THC, very little CBD, but has all of the other terpenes. And we put 20 milligrams per kilogram. It would be the equivalent of somebody eating a whole chocolate bar of a cannabis edible. The rats will eat it once and they will never go close to Nutella even again. Like 6 months later, you can put Nutella in front of them and they won't even touch it. So it really hits them hard. But we wanted to also assess whether there was neural correlates. So we did like local field potential recordings with electrodes implanted into the brain, as well as behavioral experiments and pharmacokinetics. And we see that similar sex differences as before, but with much higher plasma levels and brain levels of the cannabis, and a significant sex difference. Females have higher levels of THC, much higher levels of 11-hydroxy-THC. If you recall from the vapor exposure, they were around 3 nanograms per mil. Here we're around 50 nanograms per mil. So the edible route resulting in greater first pass, greater conversion of the parent to the 11-hydroxy, which is also an active metabolite and probably explains the long-lasting effects that you'll see on the coming slide. But we also see much higher levels of THC in the brain and the 11-hydroxy-THC in the brain, especially in the female animals. When it comes to behavior, we see long-lasting reductions in body temperature, lasting up to 24 hours, tail flick latency, which is a measure of how much pain the animals feel, lasting 24 hours, and they're showing this hypolocomotion phenotype even up to 24 hours later, consistent with some of the symptoms that are seen in kids or pets presenting with cannabis poisoning. We also see in the brain, and we wanted to see if we could have a neural substrate or neural correlate that maybe could be used as a biomarker. Maybe you could do EEG in patients that come in with poisoning to see what has changed. And what we see is that there's a massive reduction in gamma power that lasts up to 24 hours in both male and female animals. With the low gamma power, and especially in the hippocampus and the cingulate, we actually see that females already had higher power compared to the males. So you can't really see what's going on in the males, but here you can see that they also show these marked reductions that last at least 24 hours. And so we've recently published this, and now we've taken, this was an adult animal, so now we've taken it to juvenile or adolescent animals. And one of the studies, the study that showed the 1,375% increase in the US showed that the median age of the people who were being called into the CDC or the poison control was 15. And so we chose postnatal day 35 as that age. We had to pick one, so we threw a dart. But what we see is if exposed on postnatal day 35, the effects are actually much longer lasting than even what we saw in adult animals. So this is with body temperature. We see that, at around 24 hours, the body temperature starts to come back to normal, but that distance moved remains reduced up to, even 4 or 6 days later, especially the tail flick latency, which is how much the animal feels in terms of pain. We see an increase in tail flick latency that lasts up to 336 hours or 14 days. And so we're really seeing long-lasting effects of a single exposure. But what's really was mind blown for us was when we look at these animals in adulthood and then expose them again to cannabis, this time cannabis vapor. The animals exposed to the single instance of the cannabis edible during adolescence. Actually, so here you're seeing this is the Nutella group, this is the edible group. At baseline, we're seeing some sex differences between them, but no real effect remains in the edible treated groups between the two groups. Where we do see an effect is even in adulthood, the male animals exposed to the cannabis edible actually continue to show that increase in tail flick latency, which the only explanation we have is it's resulting in some long-lasting change in Pha levels. And there's at least some reports of people with variations or genetic variants in the Pha gene that don't feel any pain. And so this might be one potential explanation, but even, this is now 40 days after that single exposure, the animals still show this increased latency. But when we give them cannabis, we see that the male animals exposed to the edible and cannabis vapor actually respond very differently from the animals that never received cannabis before. They're almost sensitized or responding, instead of seeing a reduction reduction in locomotion, which we're seeing with all the other groups, these animals actually show an increase in their locomotor activity compared to when exposed to cannabis again. And we see a similar thing with tail flick latency, where they were already high. After re-exposure to cannabis, their tail flick latency increases even more. And so that single exposure now changed how sensitive they are to subsequent re-exposures to cannabis, which, we hope to continue to study to try to establish what is happening. So this brings me to the end of my talk. We are going to continue this. This is the future directions. We've now also moved to having animals self-administer the cannabis and nicotine vapor, so then they can lever press or nose poke, and they will receive the vapor instead of us just forcing them to do it. The other thing that, since with the move to Western, has come the ability to do experiments in marmosets. And the cool thing about marmosets is they have two twins 3 times a year. And so you can really do like a case control in a twin pair where everything else stays the same, but one gets cannabis or one gets nicotine and the other one doesn't. And then you can look at brain activity as well as behavior in those animals. In addition to that, on the poisoning front, we're also testing either blocking the CB1 receptor or using a negative allosteric modulator of it, or increasing the metabolism of the 11-hydroxy-THC to the carboxy metabolite to get rid of the drug sooner, to see if that would reduce at least the long-term effects that we're seeing with that single exposure. Yeah, Kristen and I were talking about it. I'm coming back in a month and I'll be giving a slightly different talk, more for a broader audience. But if you have friends interested in hearing more about edibles and vaping, we can tell them about the amazing brain. And with that, I'd like to thank all of you for being a wonderful audience, and my funding sources and the people that do the work in the lab. So thank you very much.
Great, thank you so much. So we'll now open up the floor for discussion and questions. For those of you online, you can just drop your question in the chat and I'll read them out loud. We'll start with the room, and as moderator, I will go 1 question per turn so everyone gets a chance. First one I saw was Ellen.
Thank you very much. Very interesting talk. One thing I noticed at the beginning of your slides, you had the dual exposure, and there was a peak after a certain administrative, and then there was a drop. So the longest term exposure to dual did no longer have an effect. Do you have an explanation for this?
Yeah, so that was with the preference for the condition-placed preference, and you saw it with the male adults and the male adolescents. And you actually see this, it's a characteristic inverted U-shaped curve when it comes to preference. There are doses that animals prefer, and then there are doses that, and they weren't aversive, where now the animals are not spending less time, they were just not preferring that chamber, and it was equal in terms of how much they preferred the chamber with the dual or not. And so it's because at some point the dose does become potentially aversive. And what we saw with the females, that even that high dose that was not rewarding in the males was still rewarding in the adult females. And so I think there is, and that's also consistent with previous studies where there's a rightward shift of the dose response function in female rats compared to male rats.
Thank you, Jibran. This was a fascinating talk. Thanks a lot. And I really like how your animal experience align with the technological challenges and perceptions that people have. You mentioned a couple of times animals are not humans. And I want to pick it up a little bit on the female and male differences that you showed. When you expose female animals to vaping, you don't seem to absorb the same amount of the active ingredients. Now, in clinical practice, when we see this is psychosis clinics, when we see women who are vaping, they have equally poor outcomes to men. In fact, they vape less, they smoke less in clinical practice compared to men, to show that reflects how the biological general population distribution is. But when we do, the outcomes are not any better. That's important to keep in mind.
Yeah, no, and I agree completely. And that's actually what we see with smoking as well. Less nicotine use, less tobacco use, but equally as bad or worse outcomes. And so, and there, what's really cool that you bring this up is that while not always the pharmacokinetic sex differences that you see in rats mirror those that you see in humans, but in the case of both nicotine and THC, it actually mirrors this pretty well. And so female humans metabolize more THC to the 11-hydroxy THC metabolite compared to males. Nicotine, both metabolism, and that's why NRTs are less effective in females compared to males, right? So there, you actually see consistencies in both for nicotine and THC.
So just to answer one question from our chat, today's talk was recorded and it'll be available on the Douglas Research Center YouTube channel in the coming weeks. So you can definitely rewatch and share with your colleagues there. Some question?
Thanks, Jibran. Yeah, just to jump off the sex differences question.
Yes.
Did you look at it in terms of cannabis withdrawal?
Cannabis withdrawal, we have the data, we haven't analyzed it yet. So the videos are being scored right now. Yeah. Yes.
Hi, thank you so much for your talk. So, like, I don't have any knowledge in cannabis science, so I have the doctorate knowledge, and like, we always say like, oh, if you smoke as an adolescent, then it's harmful, and but when you're an adult, then it's fine. And so you tested male, like you showed that there's a significant impact on learning in male rats when they're exposed to a teenager. But is there any study that investigated this relationship when the first consumption happened in an adult?
Yeah, and there are studies. In fact, some of the earlier studies that we did before vaping, when we were also injecting THC into animals, we actually had an adult exposure group and an adolescent exposure group. And we saw that only the adolescent exposure resulted in changes in reward learning compared to the adults. The adults did not show that change. So that's probably true. We haven't done it with this experiment, partly because of animal facility issues, but we will do it, and hopefully we'll have that data. But what's really cool, in another project that we're collaborating with Av Tremblay and Dr. Vakiralagi on, is in aged animals. And the signal seems to flip in aged animals, where THC and cannabis is pro-cognitive instead. And so there's an age-dependent spectrum as well, where, adulthood, it does nothing. Maybe in adolescence, it's worse, but in later adulthood, it might actually be beneficial.
So I'll go next. You mentioned this a few times, that just telling youth that cannabis and vaping is bad for you, or scaring them into kind of the consequences, doesn't work because they're probably going to do it anyway. So what would you recommend? Like, what's your spiel when you talk to youth? What should we be emphasizing when we do talk to them about this, and especially for those that may be using these products as a coping mechanism for other issues?
Yeah, so the first thing that I always talk about is these are all studies in animals that are wild type animals that are not a model for any genetic risk, right? So, and that's a big loss here. We're now starting experiments, and I didn't show any of the data here today, with at least humanized models of genetic variants, especially one in the cell adhesion molecule 2 gene. So hopefully we'll have that data the next time I get to come visit. But if there is any risk, any genetic risk, any familiar risk for anything like psychosis or schizophrenia in your family, then for them, that's a very different conversation. And probably abstinence is the better bet for them, right? But then for everybody else, I would say start low and go slow is a good motto when it comes to cannabis use, especially when it comes to edibles, but also to vaping and any other type, right? Like when you haven't used it before. And the good thing about cannabis compared to other things, there's almost a self-selection as well, where if you experience paranoia the first time you use cannabis, you're not going to go back to it. And it's usually the survivorship bias that people are like, I've been using cannabis my whole life, it's done nothing to me. And it's only because they never experienced any of the adverse effects, and that's why they continue doing it, right? And so I think that's another good thing that happens with here. All of them are being forced to be exposed, whereas in humans there's a self-selection. So that actually helps. So listen to your body about what that experience is the first time you use cannabis. And if you do have an adverse effect, then you let that guide you and don't try to overcome that by using more. And then be really mindful of tolerance and dose escalation. That would be the last thing, is because especially with cannabis, but with all drugs, you build tolerance, and what tolerance results in is dose escalation. And so, that dose escalation is what then potentially leads down to a substance use disorder. And so those would be the things to be mindful of there.
And that last point, I guess, is also applicable to like alcohol.
Yeah. Yeah.
Great. Okay, we have time for one last question, if there's one in the room.
So I was impressed by the single exposure and basically like the lifetime sensitization.
Yeah.
You mentioned a few times, okay, the animals are not human, or
Yeah.
that's not human. Is there some human type evidence of the same effect happening?
So the postdoc who worked on this, Richard, who's somewhere in the middle next to Jude there, he actually presented this data at the Addiction GRC meeting that just happened in Maine last month. And Anahita Basirnia, who used to be at Sinai and then now is at Yale, working closely with Cyril's group, she saw the data and then contacted me, and she said, individuals who with early life trauma compared to individuals who don't have early life trauma respond very differently to cannabis in adulthood. And they're sensitized to the effects of cannabis in adulthood. And so we're actually going to try to come up with an animal model of early life trauma and see if we see a similar effect, but also see if there are either changes in FAO or changes in CB1 receptor expression that are now contributing to this, that might contribute to both. And so that's all I know, but that was like a cool, that's the cool thing about science, is you see something and that gets you thinking of where you've seen that before, and that's how cool collaborations happen. So if anybody wants to collaborate on any of this, holler.